Hello everyone. Today we are going to study molecular biology, which is part of your subject Principles of Plant Science for first year BSc second semester. So let us discuss recap first. So we have already studied what is molecular biology, basic concept of molecular biology. So molecular biology is branch of biology or branch of science which concerns with various biological activities which takes place at molecular level into the cell. It also concerns with the various biological interactions between biomolecules like RNA, DNA and protein. So we have already studied the scope and applications of molecular biology. It concerns with every living organism, plants, human beings, animals, and all the microorganisms also. It is applicable in every field like agriculture, plant breeding, medicine, pharmacy. In every field, molecular biology is having great scope and application. So what we are going to study today is, the session agenda of today's lecture is, we are going to study DNA, basic structure, and Watson and Crick model of DNA. Also, we are going to study packaging and condensation of the DNA into chromosomes so as to fit it into the nucleus. So let us start with the structure of DNA. As you can see the diagram, this is the basic structure of DNA, which comprises of purines and pyrimidines, and in between you can see the hydrogen bonds. So let us study this one by one. So actually, nucleotides are monomers of DNA. There are two types, nucleosides and nucleotides. So nucleoside is made up of pentose sugar and the nitrogen bases, whereas nucleotides comprises pentose sugar, nitrogen base and a phosphate group. As you can see here, in one diagram or in one structure, you can see phosphate group. Okay, that one is nucleotide, and other one is a nucleoside without phosphate group. So let us discuss the components of nucleotide and nucleoside one by one. So sugar of the DNA is deoxyribose sugar. It is cyclic pentose. It is having 5 carbons. So these 5 carbons are denoted as carbon 1, carbon first, second, third, fourth and fifth. Okay. So nitrogen atom of the nitrogen base binds to first carbon and by a sugar by glycosidic bond, and phosphate group, it binds to fifth carbon by phosphoester bond. So what are nitrogen bases? These are nitrogen containing aromatic heterocyclic compounds. Okay, and there are two types of nitrogen bases: purines, in which we have adenine and guanine; pyrimidines, where we have thymine and cytosine. Remember, in RNA, thymine is replaced by uracil. So this is phosphate group. As you can see, this is the structure of nucleotide, and phosphate group is bound to the sugar by phosphoester bond. So let us study Chargaff's rule. So what Chargaff's rule is? In the DNA, there is 1:1 ratio of purines and pyrimidines. That means amount of guanine is equal to amount of cytosine, and amount of adenine is equal to amount of thymine. This is Chargaff's rule, that there is equal amount of purines and pyrimidines into DNA. So let us discuss C-value paradox. Now let's see what C-value is first. So C-value is amount of DNA in the haploid genome of an organism. Okay, so in haploid genome or a single set of chromosome, how much DNA is present is the C-value of that particular organism. So what is paradox then? So this paradox, or we can say C-value paradox, is paradox in amount of DNA and the complexity of organism. That means this is not matching. There is not a ratio that if amount of DNA is higher, that means complexity is also higher. No, it is not like that. It doesn't depend on the amount of DNA. Okay, so for example, genome of amoeba is 200 folds larger than human genome, right? Even amphibians genome is larger than human genome, but human beings are more complex than the amoeba and amphibians. Okay, they are more advanced and they are more complex. So this paradox, that means the amount of DNA doesn't mean amount of DNA is larger. That doesn't mean that that organism should be more complex and it would be higher. No, or it is more developed. No, it is not like that. So this paradox, that amount of DNA is not related to or not directly connected to the complexity of organism, it is called as the C-value paradox. So why this happens? It is because of the repetitive DNA, pseudogenes, or the introns which are present that makes that genome larger, but it doesn't make that genome more complex or more advanced. So let us discuss the Watson and Crick model of DNA, which is also B type of DNA or B form of DNA. So Watson and Crick, they have developed this model, or they have given this model by using X-ray crystallography. So see this diagram first. Now we are going to study this particular model of Watson and Crick. So yes, what Watson and Crick state that there are two polynucleotide strands, okay, twisted, they are twisted and forms right-handed double helix, okay. So what you're going to remember is the DNA strands, those are double helix, okay, and they are twisted right-handed, right? And these two strands, they runs anti-parallel to each other. That means if one strand runs 5 prime to 3 prime, other one runs 3 prime to 5 prime. That means they are anti-parallel to each other. That means 5 prime end faces 3 prime end of the other strand, and 3 prime end faces 5 prime end of the other strand, and they are helical in structure. They are twisted in the form, right? So the sugar phosphate backbones, they are outside the helix, okay. So sugar phosphate backbone, they're outside the helix, and in the core of the DNA what we have is purines and pyrimidines. So these base compositions, they obey Chargaff's rule, as we have already stated the rule that amount of purines are equal to amount of pyrimidines, right? So this in this particular DNA, adenine pairs with thymine with 2 hydrogen bonds, and guanine pairs with cytosine by 3 hydrogen bonds. So these are various dimensions of the DNA. So diameter of the DNA helix is 20 nanometer, 2 nanometer, that means 20 angstroms, and distance between adjacent base pair is 0.34 nanometer, that is 3.4 angstrom, and there are 10 base pairs per turn. So as you can do the calculation, the length of a complete turn, it would be 3.4 nanometer, that means 34 angstrom. So remember, between two bases, how much distance we have? 0.34 nanometer. So between, if the complete turn is like of 10 base pairs, it would be 3.4 nanometer. So DNA are having grooves. So the shallow groove is the minor groove, and the deeper grooves, these are major grooves. So these grooves are very important for transcription. Okay, so transcription machinery and replication machinery, those would incorporated to this minor groove and major groove. Those are recruited to this minor groove and major groove, so they can perform with the transcription. So let us discuss about the characteristics of DNA. So amount of DNA per nucleus in vegetative cells, those are constant. Okay. So C value. So as we have already discussed, that amount of DNA in haploid genome is called as C value, and it is also having characteristic of C value paradox. That means the amount of DNA, if amount of DNA is higher, that larger, that doesn't mean it is overcomplicated or it is more advanced. So only small fraction of the DNA is functional in eukaryote. That means only a few genes, they are functional and they code for protein. Other genes, they are pseudoproteins, or they are just introns, or they are silent. So DNA is a basis of heredity, and it is organized into gene, and these genes, those are transcribed to form the protein. So as we have already studied the central dogma, if you remember, okay, so what is central dogma? DNA to RNA and RNA to protein. So DNA is self-replicating, okay, it replicates to form multiple copies of DNA. Then it forms the mRNA or messenger RNA, that it copies the information of the gene into mRNA. So that is by a process of transcription, and this mRNA copy, it is translated to form the required protein for the cell. So that is why DNA is the basis of heredity, and that is why it is organized to gene so as to form the functional protein. So DNA is self-replicating biomolecule. It replicates to form DNA. It makes copy of itself, and it transcribes to form the RNA, messenger RNA, and it further transcribes to the protein. So DNA replication takes place in S phase of cell cycle. So as you remember, we have studied cell biology in previous credit. So that time we have studied cell cycle. So there are four phases of cell cycle: G1, G2, S and M. Okay, so in S phase, DNA replication takes place. So there are repetitive sequences in the DNA. These are called as satellite DNA, and these are very important in molecular studies where we identify the organism. So if any plant is unknown or any organism is unknown, we can study the DNA, satellite DNA, so as to match the previous records. So DNA can easily undergo denaturation and renaturation. That means melting and the rebinding. So when the pH is changed or when it is subjected to the alkali solution, that times it can be denaturated and also renaturated. So it easily melts and it easily rebinds. So yes, so previously people used to think that there is only one form of the DNA or one type of a DNA, which is the Watson and Crick model, which is the B form DNA. But further in discovery, we found out that there are two more or there are multiple forms of DNA in which we are going to study B form, A form and Z form. So we can compare simply this A form and Z form with the B form. So helix of the B form is right-handed. Even in the A form, helix is right-handed, but in Z form, helix is the left-handed helix. So if you study this comparatively, it would be more easy. So compare both the DNA with the B form, which we have already studied, the Watson and Crick model. So base pair per turn. So per helical turn, in the B form, we have seen there are 10 base pairs, okay? Whereas in A form, we have 11 base pairs, and in Z form, we have approximately 12 base pairs, okay? So helical diameter of the B form is 20 angstrom, that means 2 nanometer. In A form, it is 23 angstrom, and it is in a Z form, it is 18 angstrom, okay? So helical length. So helical length of the DNA is 34 angstrom in B form. In A form, it is 26 angstrom, and in Z form, it is 37 angstrom, right? So shape, see. So in B form DNA, it is not much broader or not narrower. It is like intermediate shape. It is not very narrow or not very broad. It is an intermediate shape DNA. But in A form, it is like broad. Okay, it is more wide and broad. And the Z form is very narrow, or as compared to the B form and A form, it is the narrowest form of DNA. Okay, so grooves of the DNA. Uh, in B form, narrow, deep, major grooves, these are very narrow and the deeper. In A form, those are wide, deep, and in Z form, those are flat. So as you can see the helical diameter, helical length, and the shape, those are related to each other. See, when you see the helical length is 34 angstrom and helical length is 26 angstrom and the 37 angstrom, that time you can assume the shape, and also if it is broad or it is narrow. When you compare this diameter, length and the shape, also base pairs per turn, that time you can see that okay, if it is the intermediate shape or the broadest or the narrowest. So major groove of the B form, it is narrow, deep, deep. In A form, it is wide, deep, and in Z form, it is kind of flat compared to B form and A form. So minor groove of the B form is broad, shallow. Minor groove of the A form, it is narrow, shallow, and in Z form, it is narrow, deep. So yes, remember again, these major grooves and minor grooves, these are the places where the transcription machinery attach, where various proteins, transcription factors, they attach over here. So this is the diagram for A form, B form and Z form of the DNA. I purposely have not labeled this particular diagram. Now, after reading this particular table, you are going to guess which diagram is of which form. That means which one is the B, which one is the A and which one is the Z. This is the task for you. Now next is very interesting topic, that is packaging of DNA into chromosome. Now see, So human cells DNA is approximately 2 meter. 2 meter is very large. Okay, 2 meter of DNA, and it is packaged and it is compacted inside the 5 to 20 micrometer nucleus in diameter. Okay, it is compacted where inside the nucleus, which is in the size of 5 to 20 micrometer only. So how do cell do this? That this much larger DNA, and that is 2 meter DNA, it is compacted inside such a small nucleus. How do they do it? So answer is, it is condensed. It is packaged into the chromosome and then it is kept inside the nucleus. So even the DNA is condensed, it is still be able to replicate and transcribe. There is no worry that if it is condensed and if it is very compact in structure, that it won't be replicated or transcribed. No, it is not a question. It would be still able to replicate and transcribe even if it is condensed in the condensed form. So levels of DNA condensation, it takes place step by step. So imagine wool, that how we wrap it, okay, along with one, what we can say, a ball or anything, okay, like that. In the same fashion, DNA is wrapped around the histone protein. So histone proteins are they form the nucleosome around to this histone protein. DNA is wrapped. So 146 base pair of DNA, okay, so in first level, see, 146 base pair of DNA is wrapped twice around the histone protein set. Okay, so there are histone protein set. Okay, around that histone protein, this DNA is wrapped, and this particular structure now is known as nucleosome. Okay, so histone protein along with the 146 base pairs of DNA, it is called as nucleosomes. So this gives look like bead on string model. Okay, so this bead on string packaging is termed as chromatin, which is 11 nanometer in width. So this is 11 nanometer fiber. So in second level, this nucleosomes, remember what nucleosomes are? DNA wrapped around the histone protein. So this nucleosome these are arranged into 30 nanometer chromatin fiber. That means they have 6 nucleosome per turn. Okay, so what we have first is DNA wrapped around the histone protein, and DNA is of 146 base pairs. Now we are going to arrange this nucleosome into 30 nanometer chromatin fibers, and in which we have 6 nucleosome per turn. And this particular structure is now called as, or this particular arrangement is now called as solenoid fiber. So now in the third level, chromatin fibers, those are arranged in the loop. Okay, now what we have is we have loops, loop domain, each approximately like 120 kilobases long, and it each comprises of the 6 loop. Now in next higher level, this 120 kb DNA links to the rosette form, okay, and which are attached to the nuclear matrix. Now this 120 kb DNA, they forms the rosette and they are attached to the nuclear matrix. The last and the final step where the DNA is condensed, it is like form chromosome. Okay, the final level of compaction of the DNA is to form the chromosome previous to mitosis, and the diameter of the chromosome is 700 nanometer. So that means this 2 meter DNA is finally compacted to form the chromosome by condensation. So assignment for you is, you are going to write in brief about Watson and Crick model along with the diagram. Okay, and you are going to send me this assignment on my mail ID, which is shwetaburade@ktshmcollege.ac.in. So what is summary of today's session? So we have studied the structure and types of DNA. So see, DNA is a basis of heredity. Okay, it carries the information, carries the genetic information from generation to generation, from parent to offspring. So to study the DNA is the most important part of molecular biology. So when we study DNA, the next level is to study RNAs and proteins and interaction between the DNA, RNA and the protein. So we have also studied characteristics of the DNA in this particular lecture. So in next lecture, we are going to study types of RNA and replication of DNA. So why we are going to study types of RNA is, we are going to study mRNA, rRNA and tRNA. So these all three types of RNAs, these are involved in transcription. Okay. So to study further molecular biology in next years, so as to study transcription and translation, you are going to study the three types of RNA: mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, which is ribosomal RNA, which forms the ribosome. And in next lecture, we are going to study replication of DNA, semi-conservative replication of DNA. That means how DNA makes copies of itself, okay, how it replicates. So I hope you have understood this particular lecture very well. Thank you so much.